Started. Thanks for braving the elements and coming to see Dr. Jennifer Lachowiec. That's how to pronounce it. I remember that very clearly because she said it's like I have no wits. I have a lack of wits, but which you will see is clearly not true. She grew up in Indiana. She was a competitive swimmer and a fantasy book reader. She completed her BS at the University of Wisconsin, where she majored in genetics and anthropology, and she did an Ironman triathlon. Right out of college, she moved to Seattle for her graduate studies at the University of Washington. She graduated in 2014 with a PhD in genetics and plants. For her postdoc, she worked on genomics and evolutionary genetics and started writing cyclocross, and that was in Michigan, right? Mm-hmm. In 2017, she started her lab here, integrating her genetics training with the ability of plants to withstand changing environments, genetic mutation, and randomness of development. And her next sport still awaits, but I'm pushing for skiing pretty hard. So welcome, Jennifer.
Thanks, Ryan. Okay, so I think that this is a really cool image. It's called Capturing Chaos. And so today I'll be talking about harnessing noise to manipulate biological systems. So one thing that I think is really interesting is when you see a set of identical twins, there are minor differences among them. So when you think about how identical twins emerge, you have an embryo, and for some reason, some of the cells break off and develop into two individuals, and they share the genetics, at least their initial genetics. But even though you meet a pair of twins and you're like, oh shoot, how am I gonna know who is who? You look, you decide, and you're like, okay, I think I got it. Then you meet them again, and they're wearing different clothes, and you can't remember who's who. So I think it's really interesting to think about how during development, noise can arise that influences how development proceeds. And we can even see minor differences in really young children before they've had lots of time to accumulate environmental differences and so forth. So when we think about the differences we observe between individuals, this variation is driven by multiple sources. So we have differences in the environment, we have differences in the genetics of individuals, but we also have differences that arise due to stochasticity or noise. So the system that I study are plants in general, but I work a lot with the model plant Arabidopsis. And this is an example of where changing environments or different environments can influence a phenotype. So these two plants are genetically similar, but the one on the right, was grown in shade. The plant is trying to find light, so it has this spindly growth to try to reach light. And so that is an example of how the environment can influence the process of development and growth and introduce variation. On the other hand, across different populations of Arabidopsis thaliana, we also have genetic differences. So these are the rosettes that are the exact same age across different populations of Arabidopsis. So you can see that there can be a range in size. And this again arises due to genetic differences. All these plants are the same age and were grown in the same environment. One thing that the topic of today, what I'm going to discuss is a third way that variation can be observed in phenotypes, and that is stochastic variation. And I'm sorry if the title of this talk misled you and you thought I was going to be discussing acoustics and noise to manipulate populations, because today we are talking about stochasticity and variation that arises due to random events. So what do I mean by this? So stochastic variation comes by many names, including air or noise, and I'll have more of them later. Many different fields study concepts like this, but kind of name them in different ways. So what I've pictured here are many seedlings that are all the same genetic background. They're all grown in a highly controlled environment, and you can kind of see in the background, they're in a petri dish. So we have this really uniform substrate in which they're grown. They're grown in a highly controlled growth chamber. However, there are all sorts of differences among these, despite the fact that they have grown up in the same environment and share the same genetics. So this is kind of what people would call a type specimen or a wild type looking plant. We have the cotyledons, the initial embryonic leaves, and the formation of 2 adult leaves. But as we look across these individuals, we see variations. So here we have these strange looking adult leaves that are pointy. Over here, we're missing an adult leaf altogether. We have variations in how the leaves are arranged, about the center stem, and so forth. And so all of these arise due to just random variation. And I'm going to talk a little bit about what that random variation can be. So this random variation that occurs can affect any kind of trait and result in phenotypic noise, not just what you see in seedlings, for example. So you can quantify noise in adult traits, and for this example, I kind of have the height of plants, the width of the rosettes. We can look at traits that are discrete, so like the number of petals. We can look at the hue of leaves, but we can also look at more complex traits and measure their noise. So for example, we can assess noise and how a plant responds to an herbivore, or we could assess noise in things like growth rates. And I have some projects where we're looking at noise in the circadian clock. So these are all, for every trait, you can essentially measure variation that's due to noise. This noise that affects organisms can affect any organism. So across the tree of life, there are stochastic events that influence how individuals grow and develop. Now, if we're thinking about single cellular organisms, you may argue they don't have development, but you can also have noise in the metabolic processes occurring within them. So development is stochastic and not deterministic. So to say that, how does stochastic variation arise? There's kind of 2 ways that you can think about it, and both are very important. So the first is that as an organism develops, cells divide in the multicellular framework. So what I have pictured here is a single cell, and the green circles are supposed to represent some important molecule to that cell. Because of diffusion, it might be that the number of molecules that end up in the daughter cells can vary among individuals. And so what can happen is we have differences among these daughter cells. Interestingly, there's evidence that these kinds of divisions can influence what these cells become. So what I have here is a cartoon of a root. And along a root, we have root hairs that develop. And it turns out that the number of a particular molecule present within a cell defines whether the cell of the root becomes a non-hair cell or a hair cell. And so these stochastic noise differences due to molecules moving between cells, or how many molecules end up in a cell, can influence the process of development. Through this process of development, we essentially have an array of phenotypes when all else is equal. So starting off with a single genotype, a single environment, we still can get variation due to the process of development. The second source of variation that I would consider stochastic is microenvironmental variation. So even in the most highly controlled conditions, so some petri dish with a consistent substrate, or in a highly controlled growth chamber, we can still have microenvironmental differences that are really difficult to control. So this can also lead to stochastic noise. In addition, you can imagine, like out in the real world, that you have all types of environmental variation that's difficult to control. So even within a small plot, you may have, or you do have, environmental differences due to the presence of other organisms or soil composition and so forth that influence how development occurs. So these together, both the error that we have occurring during development, as well as microenvironmental variation, contributes to the stochastic noise we see. It's difficult to parse these apart And there's basically some beautiful, elegant experiments in bacteria where they've actually been able to pull apart air due to stochastic noise in development versus external noise due to the microenvironment. However, these are done using like transgenics and high throughput microscopy. So maybe not super accessible to someone working in plants. You're looking at noise. So why does phenotypic noise exist? And I kind of think about it in two flavors. The first, I dub the second law of thermodynamics, and the second is the process of evolution. So in the second law of thermodynamics, one aspect of it states that there's a natural tendency for any isolated system to degenerate into a more disordered state. And essentially, I feel like this captures the process of diffusion, underlying these stochastic differences between cells. Evolution, however, has also tuned the degree of phenotypic noise we see in traits. So in order for evolution to have this effect, the traits that we examine, like noise, needs to be inherited, and it turns out that noise is inherited. And I think one of the neat ways to think about this phenotypic noise in terms of an evolutionary context is BetHedging. So I think there are probably some people here who know a lot more about BetHedging in weeds than I do. But essentially, conceptually, BetHedging is a concept of don't put all your eggs in one basket. And a good example of this is seed banks. So when plants, certain species of plants have evolved mechanisms multiple times to delay germination to increase the chance that they reproduce in favorable conditions. So there's a lot of work on this, studying plants in the desert where there's rare rain, but also examples of this across the tree of life as well. So seeds from the same plant, so offsprings of the same plant, those seeds may germinate, grow, and reproduce. They may die without reproducing. Alternatively, they may delay germination and try again in another year. And so this is the bet hedge. Essentially, by entering a dormant state, individuals can endure conditions that are suboptimal for growth and reproduction, thereby increasing a population's long-term geometric fitness. So a lot of studies of bet hedging are ecological studies done in natural environments. So you can imagine that it would be difficult to parse apart whether the differences in bet hedging rates are due to things like environment. So is the fact that the seeds are dormant due to a poor year, causing more seeds to be banked? Or is it due to different genotypes of plants banking seeds? Or is it possible that these seeds just stochastically germinate? And so parsing that apart in a natural system is difficult. So in order to really focus on noise specifically, we need to try to account for or eliminate the environmental contributions to phenotypic variation, as well as the genetic contributions to phenotypic variation. And so one way to do that is just to look within a specific individual. So one individual kind of mostly shares its genetic information, and across particular traits should experience similar environments. So quantifying noise in this way has several names again, and some of those you might have heard of include fluctuating asymmetry or developmental stability. And what people can do is take their trait of interest, like a leaf, and measure how symmetrical it is. And there's actually a journal of symmetry where you can see tons of studies that start to address this. Leaves are, I think, a really nice example. There's even some data that shows asymmetry actually improves photosynthetic rates in plants, which was interesting, but I think not fully understood if it was true or not. Another kind of asymmetry that you could measure, for example, are the angles that leaves are added onto the stem of a plant. So there are predictable angles that we expect leaves to be added on. So for example, there are some plants in which the leaves are added opposing one another, so 180 degrees apart. Others where they're added 90 degrees apart, and others where they're added at approximately 137. So we can look at an individual plant and measure each of those angles as a spiral of leaves is added, and look at the variation in this angle. And that would be an example of capturing noise. And so down here, for leaves 1 and 2 in this particular example, you can see that there is a spread in angle. And same for leaves 2 and 3, and for 3 and 4. And in this case, they're looking at a mutant in which the angle is the angle is greater in this particular mutant. Another thing that has been observed is that there may be some information about sexual selection and fluctuating asymmetry. So there's data that shows that animals and humans prefer mates that are more symmetrical. And so there are studies where people essentially take 3D scans of one another's bodies and faces and assess how things made. This has also been done with like deer antlers and information about that too, which I thought was pretty interesting. And when people, hold on, I'm gonna describe this poorly. So for example, if hunters submit these giant antlers that they've found, the symmetry is accounted for in the scoring that it's given as being a champion trophy kill. So we also prefer symmetrical in the animals that we consider trophies. So fluctuating asymmetry and developmental stability are kind of looking at noise within a single individual, but we can also look at noise across a population if we can eliminate genetic variation. And a lot of plants can be inbred so that essentially all of the offspring are genetically identical, like little clones of one another. And so you can measure a trait in many of those individuals and capture the variation in that trait. And this kind of way to describe noise is often called robustness or uniformity. And so this example here, I have 2 different genotypes. There is the mock genotype and then the mutant genotype there. And I measured the embryonic stem length of these individuals. And in the mock or wild type individuals, I have this pretty tight distribution centered around 19 millimeters. Whereas in the mutant, there's a much greater spread. And even if you look at that distribution, it seems to have like a little hump somewhere. And so we have differences in the noise of this embryonic meat. And we capture this by looking across the population. The one difficulty, the greatest difficulty? Nah. One of the difficulties in studying noise is the fact that you need high replication. So oftentimes you can estimate the mean of a population without looking at too many individuals. But if you want to capture the variance of a population, you need to look at many more. And so this is a simulation we did estimating if we wanted to look at the coefficient of variation, which is the standard deviation divided by the mean, so like a normalized variance measure, how many individuals we would want to look at to kind of decide whether we had enough certainty in our estimate of the noise. And generally, the more the better, obviously, but it's much more than you would need to just capture the mean. So today, to get at really the title of the talk, how can we harness this noise? How can we study it? And I argue that one way that we can study noise so that we can harness it for our benefit is to identify the genes that influence noise and unraveling how they work. The particular stories that I'm going to talk about are inspired by the corn whisperer, Drew Haynes. So Drew Haynes is the winner of the 2018 National Corn Yield Contest. He's from Maryland, and this was his 5th year participating in the contest. This is a contest that is hosted by one of the national corn organizations. And this competition is for farmers to produce the most bushels they can off a single acre. And so farmers like Drew agonize over every detail of their field. They figure out which parts of the field they should apply more nitrogen. They figure out what the soil condition is across like every few square inches and figure out how they can maximize yield. And so one thing that's been noticed across the winners of this contest is that emergence is key. And this is for corn. We don't know what other organism it is, but basically what this is saying is that we need to ensure that the corn emerges within a short time frame so that they're all growing in sync. And one way to do that is to make sure the soil temperatures when planting are a certain degree. But I argue we could think of this corn emergence as potentially a form of noise. Do we have all the individuals coming up at the same time, or are they spread out? Do we have variation? So are there ways that we can improve our understanding of noise in important traits like emergence and see if we can use this to breed crops in certain ways, or perhaps even find chemicals that we can use to manipulate the processes underlying this. So to study noise and plant emergence, I mentioned that to study noise, we need to minimize our genetic variation and minimize our environmental variation. So to minimize environmental variation, we want to make sure we do our studies over experiments that have the same treatments. We would potentially want to focus on small plots so that we minimize spatial variation. And we also want to look at genetically identical individuals. And in crops, or in cereal crops, this is pretty straightforward because hybrid corn, they're all clones of each other. They're all the same. With inbred wheat and barley, again, they're very genetically similar. So with these, by taking these approaches, we can minimize noise due to other sources. So ideally, one would use remote sensing to get a really high level of replication across fields and focusing on means of variation per small plot. So this is one thing that a student in my lab is working on. Another aspect that we're trying to build is automated phenotyping in the lab so that we can watch the plants grow and capture rates of emergence. And the way that we're doing this is by using brambles of Raspberry Pis. So Raspberry Pis are $5 to $30 little computers about the size of a credit card. And you can add on componentry to these little computers. You can put on a SD card reader. You can add an Ethernet cord port. You can put in a camera. And so what we're doing is essentially mounting several dozen cameras within a growth room at the plant growth center so that we can watch every single plant grow, which is about 1,500 simultaneously in the room. This is ongoing. We haven't built our bramble yet, but we're working on it. So you may imagine, meanwhile, our approach for phenotyping noise is through brute force. So my colleagues in the lab have invested a lot of time measuring plants to capture emergence. And so the ways that we're doing this is by studying the plant Arabidopsis thaliana, model plant. And Arabidopsis is a genetically variable plant across the world. It's found in temperate climates across the world, and thousands of populations have been identified, and many of them have also been sequenced, so we know the genetic makeup. And so what we're looking at are some proxies for emergence that are easy for people to measure. So one of these is the embryonic stem length. So essentially we take a Petri dish, we place the seeds on the dish, place them vertically in the dark and let them grow for several days. And so we measure how long these stems are after a week. So you can imagine this is similar to putting a seed into the ground and it's going to grow up against the gravity force. Another proxy that we're using for emergence is Arabidopsis seed size. So there is some good correlation between the size of the seed and the length of the embryonic stem. So we're also measuring seed size. And this is a little bit easier because there are good automated ways to just take a picture of a bunch of seeds and it will measure how big each of them are. And so we're able to capture noise that way. And so our goal is to look at these phenotypes and capture some kind of standardized dispersion of the phenotypes to measure noise. And one of them that we're using is called the Fano factor, which is over here. And this is the variation divided by the mean. So we can account for differences in means across different genotypes. So what we've done is we've measured the Fano factor for embryonic stem length across about 75 different populations of Arabidopsis, so these worldwide populations. And again, they're genetically similar. And so we are able to get a range of Fano factors. So on this end, we have plants that have very consistent embryonic stem lengths, whereas over there, they're very inconsistent. And each of these box plots is actually 3 Fano factor measurements. So you can see we do have some variation in our ability to capture noise as well. But when you run some quantitative genetics math, we can see that this is highly heritable, the Fano factor or noise. So what we hope to do is to find genes that underlie this variation in noise across populations. So we're using the approach of a genome scan. And so in a genome scan, essentially, we're looking for an association between a particular site in the genome and this trait of interest. So in Arabidopsis, there are 5 chromosomes, and this is their relative sizes. And for every single one of these populations, we know what base they have in the genome. So what we're looking for essentially is a site or a base in the genome that is different between the populations, but associated with the Fano factor. So let's say that we have a base here that's either A or T. If we end up seeing that the Fano factor tends to fall in the A group and it's low, versus the T group has a higher Fano factor, that would be a really strong association and suggest that this particular base underlies this trait. Then we can look at what gene this is and start to explore what mechanism might underlie the production of noise in this trait. So this is the output of doing this. So essentially, every base across each chromosome, we check to see if there's an association with the Fano factor. And the smaller the p-value is of the statistical test, the higher the point. So this is the minus log p-value. And if we pass a threshold, which is like a multiple testing correction, we have potential candidates that we can study further. And so this display is called the Manhattan plot. And what you can see is there are some peaks that are heading towards significance. So especially these ones that are supported by multiple dots. These are potential candidate genes that underlie noise and embryonic stem length. So the Fano factor, you can see, is comprised of the variation divided by the mean. So you might have some concern that, well, what if these are just genes that control the means of traits and nothing else? So we also do genome scans for the means as well and compare and see if we find the same sites. What you can see is that the pattern of points and where they're located is not matching this very well. So at least the major contributors or the major genes that contribute to noise don't necessarily also contribute to the mean. So again, we're using this approach to try to find interesting genes to study, but we are also trying to find different statistical ways to measure noise in a population. So I've been talking about the Fano factor. You can also use the coefficient of variation, but another thing that is interesting is that neither of those capture the fact that there's actually a shape to this distribution. Is that shape interesting? It might be interesting if it were bimodal, especially if you're looking at something like dormancy in seeds or something like that. And so we are also using approaches where we can basically measure the differences between populations using a type of distance and using multivariate analyses to describe a population with a single unit. And these kinds of approaches are developed, or the ones that I found at least, have been developed for looking at distributions of single cell data in bacteria or in human cell lines. And so I also just want to show that we are able to use some of these multivariate metrics to also identify potential sites in the genome with genes that may influence these distribution shapes as well. So some of you may be curious about what some of these mechanisms are. And at this stage, I don't feel like I've validated these genome-wide approaches enough to tell you about them. But I do want to tell you about some previous work that I've done looking at a few mechanisms that influence noise. One mechanism that influences noise. So it turns out that emergence is influenced by the action of many plant hormones. So plant hormones influence when the seeds are going to germinate and how the initial seedling elongates. And so some of these hormones include gibberellins, auxins, and brassinosteroids. And so I had been looking at brassinosteroids for a completely different project and noticed that there seemed to be some effect on noise. So just to let you know, brassinosteroid signaling is comprised of many components. Essentially, brassinosteroids they're steroid hormones. They're similar shapes to testosterone, estrogen, and so forth, but plants have their own version. And these steroid hormones bind to a receptor that's at the membrane of the cell, and this goes on to signal downstream to proteins that influence what other genes are turned on and off. So I had been looking at some of the genes involved in regulation in response to brassinosteroid signaling. So down here in the pathway. And what I found is that one of them, named BEH4, when I looked at a mutant of it, I had a high increase in noise, again in the embryonic stem length compared to wild type. So it seems like I need this gene to function properly to have a wild type level of noise. This signaling pathway here at this stage has 6 different genes that are involved, and they're related to one another. So the gene that I just showed you is way back here, BEH4, but it has 5 other relatives to it. And so I asked, well, do all of these have the same effect? So I measured the noise in the embryonic stem length again and found that only BEH4 of these 6 seemed to have this effect on noise. So I was wondering what's going on here. In the literature on trying to understand noise at a molecular mechanistic level, people frequently talk about redundancy. So redundancy is an important aspect of providing robustness to mechanical structures and electrical grids, for example. So in this picture here, I have an example of people adding more trusses to provide redundancy and improve the robustness of a bridge. In addition, you can see in this electrical grid, there are many different pathways to go from one point to another. So there's redundancy in the way that information can travel. So because this gene family that I was looking at has 6 members, I wondered, well, will I keep getting more noise if I remove more of them? So if I keep removing pathways that can produce the trait of interest, do I have worse noise? Turns out that I didn't. So when I removed a second member of this family, the noise actually reduced. So it seems like in this particular example, with this particular gene, redundancy is not where noise is being maintained. There are other features here that are important that may influence the noise in the system, and one of them is the topology. How are these different components connected to one another? Is there something about the way they're connected that provides reduced noise or robustness to the system? And so what I did is I figured out the way that these 6 different genes are communicating with one another and built a topology. So here I know now that BEH4 positively regulates BEH3, 1, and 2, whereas it's negatively regulated by BEH3. And so I had this topology about how they're talking to one another. So currently, to try to study this more further, I'm working with a mathematical computational biologist at University of Delaware to model this dynamically to see if we can understand if it's the shape of these connections that influences the noise of this trait. But so far, that is unclear. So to wrap up, these are the takeaways I want you to go with. First, development is a stochastic process that produces an array of phenotypes, all else equal. The degree of noise in a phenotype is influenced by genetics and the environment. And because of this heritability of noise, evolution has tuned noise levels. The genes that contribute to noise don't necessarily affect the mean of a trait. And noise is an emergent property that is affected by the underlying network of genes that contribute to traits. And lastly, I think that if we have increased understanding of the genetics underlying noise, we might be able to manipulate it for our benefit. So these are some ways that I'm thinking about that. We might be able to manipulate the noise of traits without affecting the mean. We might be able to, for example, improve consistency of emergence in crops where that's deemed to be important. We might be able to reduce entrance into dormancy in weeds and so forth. You can probably imagine more. So with that, I just want to thank you for your attention. Again, I said we are taking a brute force approach right now in the lab for measuring. So I have lots of great undergraduates in the lab. And I'd also like to thank the Center for Faculty Excellence at Embry for funding some of the early studies and looking at noise in this way. And I'd also like to recognize my PhD advisors lab where I was working with BEH4 and the folks there. So thanks for listening, and I'll take questions.
Okay, I'll ask a dumb one. If you remove BEH4, do you just revert to the mean?
No. So when I take away BEH4, the mean is actually a little less than the wild type. So BEH4 affects both the mean and the noise. Yes, great question. There.
So presumably the noise genes are gonna be, don't have noise genes that all the noise for everything.
There are some genes that have pleiotropic effects on noise. So there are some genes that influence the noise of many traits. And generally, those are genes that are involved in very essential processes in the cell. So genes that are involved in maintaining protein functions, for example. So there are examples where it's a single gene has many effects. In terms of manipulation, I don't know if those would be the best ones to target because they might have positive effects at this part of a plant, but negative effects somewhere else. Yeah.
In your concluding statement, you said that you might be able to reduce weeds entering dormancy. In other words, you're going to try to damage the weed by not letting
And then maybe then you could spray.
it.
Then maybe you could reduce the seed bank potentially. This is an idea. I think it's something to explore and consider as something to think about. But it's definitely half-baked. Yeah.
You mentioned the pedal number is influenced by some of these brassicacea.
Yes. So there's this really nice paper by Angela Hayes' lab where they look at one species of brassicaceae. I forget which one. It's hirsuta, I think. See hirsuta. But they actually, that particular species varies in the number of petals from 0 to 4. And they actually find which gene in that species is causing the variation in petal number versus that like consistent phenotype you see in other populations. Yeah.
In brassicaceae reduction.
0. Yeah. It's a really nice system. She has a really nice system. Yeah.
But in that example, those were QTL that controlled leaf number, not noise QTL, right?
So this was across populations of this particular species. You can find populations that make no petals, 1 petal through 4. And so they were kind of trying to figure out why in this species can you have variation across populations, whereas in other species you have this extremely consistent invariant trait. And so they were able to find some specific genes to do that. And they didn't use QTL, they actually used a candidate gene approach. So they had some guesses of what genes that could be, so they checked and they were right. I mean, they might have also screened like 50 other ones, but they don't put that in the paper.
So we do have a question from online.
Yes.
And Greg says, in the last example of BH4, when you remove alternative pathway, noise is reduced. So kind of redundancy is fully contributing to noise, just in the opposite way you were thinking. In other words, by not giving as many signaling options, the variation of noise is reduced.
Yeah, so I haven't really explored why the noise was going down as I removed more components. Yes, it could be that there was kind of this canalization where you have to force all the signaling to go through a limited pathway. And I think that that's a really good point, that you could reduce noise by removing possibilities. That's a great question.
Can I follow up on Matt's question? I'm going to say the mustards are very regular with 4 petals, aren't they? And so this is really something to be able to control that.
But they do know some species naturally.
Excuse me.
Yeah, well, the other thing that I've also been kind of daydreaming about is through the process of domestication, have we changed noise of traits? Like, have we inadvertently or vertently changed that traits seem to be more stable? And I don't know how to study that because we have genetic variation, environmental variation, and it would be difficult to control to pinpoint it to noise, but that's something I've wondered about too. Because if evolution can tune noise, we've probably done it as well through domestication.
Do you want to speculate? Yes or no?
I want the answer to be yes, but I don't know. I have to think more about it.
We've increased it or decreased it?
That we've decreased it.
I'm going to bet against you.
What is your question?
I had a question about one of your early slides had the statement that development is stochastic and not deterministic. And I guess I'm wondering is it really an either or?
No, that's true. It's a matter, it's of course, like we have a development following a pathway. So yes, I think that my statement was definitely on the extreme end. Yes, matter of
like a layer of a stochastic thing
on top of.
some underlying.
Absolutely, absolutely, yes.
for stochasticity is greatly increased.
Yeah, and people are finding more and more that certain developmental decisions must occur through some of these noise-related decisions. And so that's a kind of a newer kind of exploration in terms of looking at different cell types forming and things like that. All right, well, thanks everyone for coming.
That is so